Apparently. Test, test, test. It is time to start.
Let's remember Meredith. She's having her surgery now. So, oh, Jess. Let's remember Meredith, and don't forget the quiz this Friday is over chapter 14. Also remember there is a natural science seminar. If you wish to come, please tell Mrs. Schaefer. We can only have, I think it's, what, 50 people tops, or maybe less than that, in a white auditorium. And if you have questions, submit them beforehand because we can't pass the microphone around. So if you have any burning questions about COVID-19, ask Tom Kunstman. Yeah. 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 It'll be recorded. All right. So more about that when Friday comes. Let's begin. Lord, we lift Meredith up to you. We ask you that the surgery goes well, that you'd help her withstand the anesthesia well and not have any adverse effects because of it. Help her to heal quickly. Help her to get back on the golf course quickly. Thank you for these students here. We pray for Dane and his colleagues as he rides to a soccer game. Help them get there safely. Help them play hard, play well, play honorably. Bring them back to us safely. Thank you for these students here on this cold day. In Jesus' name, amen. Boy, we went from 70 to today. It feels like 20 out there. My word, I know it's not 20, but it feels like it. Okay, gluconeogenesis. We take 7 of the 10 reactions of glycolysis and we reverse them. Of the 3 that are irreversible, we have bypass reactions. So the first is fructose 1,6-bisphosphate. That will convert fructose 1,6-bisphosphate into fructose 6-phosphate. Then phosphohexadiisomerase will convert fructose 6-phosphate to glucose 6-phosphate. In many cells, it ends right there. If you want glucose and you want to make glycogen, you want to use it for something else, you can add a UDP group, use it for biosynthesis, whatever, that's where it ends. In liver, however, if the cell wishes to secrete the glucose, it can't secrete a phosphorylated molecule. It takes too much energy to do that. So it locks off that phosphate using glucose 6-phosphatase and water. And it makes glucose and inorganic phosphate. This can then be secreted into the bloodstream. That's what raises your blood sugar in the post-absorptive state. This is, of course, under the control of the hormone glucagon. Glucagon will increase the activity of this enzyme, increases both the synthesis and the activity of the enzyme. And the forward reaction, of course, is going to be hexokinase. Forward reaction here, of course, is going to be phosphorexokinase 1. Now, this bypass here is right a bit more different. Going from pyruvate to phosphatidylpyruvate is quite energetically demanding, so it's broken into 2 different bypass reactions. It takes 2 bypass reactions to get you from pyruvate to phosphatidylpyruvate. So as you said before, fructose 1,6-bisphosphatase, that is a gluconeogenic enzyme. When that enzyme is active, PFK1 will be largely inactive. When PFK1 is active, this enzyme will be largely inactive. They show reciprocal regulation. For the formation of phosphoenolpyruvate, you first have to convert pyruvate to oxaloacetate. Oxaloacetate. Now this requires a carboxylation. all carboxylases use as their cofactor biotin. Biotin. So this enzyme, pyruvate carboxylase, is a biotin-containing enzyme. And it converts pyruvate into oxaloacetate, and it has to burn an ATP to do it.
Do you know the mechanism for that? 'Cause
Yeah,
that is so hard to put that.
it's really
like, put it
interesting.
on that part
You
that-
phosphorylate the biotin to make phosphabiotin, and then that is easily displaced by the bicarbonate. And then the biotin is on a lysine, and it has a long aliphatic chain. It swings back and forth. It swings one end, gets phosphorylated, swings the other end, and it swings back, and it sticks the, because this is a bond to a amino group, so it's an activated bond, and it swings up and sticks it onto the methylene. This hydrogen gets abstracted by a histidine, and so it becomes a carbanion, and then it attacks that, and it's yeah, a very interesting mechanism. Pyruvate carboxylase is a mitochondrial enzyme. It's found in the mitochondrial matrix. There is a transporter that transports pyruvate into the mitochondrion in exchange for a proton. So pyruvate gets into the mitochondrion very easily. Pyruvate carboxylase is a tetramer. It's a homotetramer. And you can see the tetramerization domains composed of these beta sheets. Those beta sheets are going to help with the tetramerization. Virtually all pyruvate carboxylases that have been examined are homotetramers. Very common feature of the enzyme. After it carboxylates pyruvate to make oxaloacetate, it needs to convert oxaloacetate to phosphatidylpyruvate. This is a very high energy molecule. You have to burn another ATP to do that, except it's GTP. Now, this enzyme, PEP carboxykinase, comes in two isozymes. One uses GTP, the other uses ATP. One is mitochondrial, the other is cytosolic. The mitochondrial enzyme tends to be cytosolic because GTP is higher in the cytosol than it is in the mitochondria. Yeah. There's two versions of this enzyme. You know what? I take that back, that's wrong. The bacterial versions of this enzyme use ATP, not our version. Sorry, that's wrong. The bacterial versions of PEP carboxykinase use ATP. Some do. Our carboxykinase, PEP carboxykinase uses GTP pretty universally. So in liver, there is a PEP carboxykinase both in the cytosol and the mitochondrion. And the cytosolic form is controlled by hormones because it's important for gluconeogenesis. Cortisol in particular is the hormone that controls it. The yeast version of this enzyme and the bacterial version of this enzyme show 50 to 60% amino acid identity. That's pretty high. So these enzymes, they're pretty closely related. So as you can see, it's going to take the gamma phosphate here, and it's going to transfer that to here. And these electrons are going to come here, make a double bond. You make phosphoenol pyruvate that way. The CO2 then takes its electrons and leave. What kind of a leaving group of CO2? Quite good, quite good. It's very stable, quite good. So it takes a 1-2 punch if you're going to get pyruvate to phosphoenol pyruvate. You have to convert it first to oxaloacetate. You have to convert it then, oxaloacetate to phosphoenol pyruvate. Both steps require NADP. Gluconeogenesis is an energetically expensive process. Absolutely vital for cells. Now, as you might guess, this process is quite heavily regulated. So PEP, carboxykinase, is activated by acetyl coenzyme A. Now we will look at the fate of pyruvate in the mitochondria. And it encounters a gargantuan enzyme complex called the pyruvate dehydrogenase complex. Very complicated, has a very complicated mechanism, but this enzyme shows end product inhibition. However, that end product actually activates pyruvate carboxylase. So again, what inhibits one phase activates the other. You know, this direction, a buildup of acetyl coenzyme A inhibits you. You want to go the other direction, buildup of acetyl coenzyme A activates you. So again, reciprocal regulation. Now, the plot thickens because mitochondria don't have good ways to export oxaloacetate into the cytosol. Oxaloacetate is the first product, it's the first substrate for the Krebs cycle. So the mitochondria protects it somewhat jealously. But you need oxaloacetate to carry out gluconeogenesis. And pyruvate carboxylase is found in the mitochondrion. It's not found in the cytosol. So what's the cell to do? The cell is going to use a series of shuttles. Shuttles that will shuttle molecules from the cytosol to the mitochondrion and back again. So pyruvate and phosphoenol pyruvate are easily shunted from the mitochondrion to the cytosol. So these reactions with phosphoenol pyruvate, they can occur either in the mitochondrion or they can occur in the cytosol. Also because there's two isozymes of phosphoenol pyruvate carboxykinase. So if you make PEP in the mitochondrion, no bother, you can send it straight to the cytosol, not a problem. Pyruvate can come in to the mitochondrion, there's no problem with that. There's a pyruvate transporter that will do that. Then it will encounter pyruvate carboxylase. Now, if the oxaloacetate encounters a mitochondrial version of phosphoenol pyruvate carboxykinase, then oxaloacetate gets converted straight to phosphoenol pyruvate. It goes to the cytosol. You pass go, collect $200 and perform gluconeogenesis. Not a problem. However, there's another way to skin the gluconeogenesis cat. That is, you take oxaloacetate, you reduce it to malate. You reduce it to malate. This is useful because reductant tends to be abundant in the mitochondrion, tends to be relatively sparse in the cytosol. So this is a way to pass reducing equivalents to the cytosol from the mitochondria. So you reduce it using malate dehydrogenase. That's a Krebs cycle enzyme. It's found in the mitochondria. But that enzyme also has a cytosolic lysozyme. And it's used for various purposes. One of which is to give reducing equivalents back to the cytosol. Then you oxidize it to it being malate to oxaloacetate, then you have a cytosolic phospholipyruvate carboxykinase, then you make phospholipyruvate, you perform gluconeogenesis. So either way, Bob's your uncle, there you go, you make glucose. Yeah, that's if you want to shunt reductant to the cytosol, if the cytosol needs reductant. So for example, if it's going to make lipids, going to make fatty acids, has to have reductant. It's going to make glucose, needs reductant. So these processes that occur in the cytosol, they need reductant, but NADH can't pass through the mitochondrial membrane. So what you do is you reduce organic compounds, you shunt those across the mitochondrial membrane. Then you unload that reductant in the cytosol. That's how you get reductant from the mitochondria to the cytosol.
That pathway gets activated when NAD.
Yep. Yep. Precisely. So the shunts, essentially, they will ensure that your metabolites formed in the mitochondrion are available to the cytosol for gluconeogenesis. Because remember, gluconeogenesis, like glycolysis, occurs in the cytosol. It occurs in the cytosol. After this, everything's going to occur in the mitochondria. But this occurs in the cytosol. And the concentration of these molecules required to build glucose molecules tend to be low in the cytosol. So shunting them from the mitochondrion, where those metabolites tend to be a right bit higher in concentration, is quite important. And these shunts, this so-called malate phospholipyruvate shunt, is really quite important. Now, if you make glucose 6-phosphate, or for that matter, if you make fructose 6-phosphate, you can use those for biosynthesis. If you wish to make starch or glycogen, you have to convert glucose 6-phosphate to glucose 1-phosphate. That's okay. We have an enzyme that does that, phosphoglucomutase. Very widely distributed, ubiquitous enzyme. Now for starch, you're going to use the substrate ADP, glucose synthetase. ADP glucose synthetase, because starch synthase will use ADP glucose. You don't need to use ATP then to make starch, because this bond is already activated. You can stick it right in and you make starch. That's cool. So starch, you use ADP glucose. For glycogen synthesis, that is in animal cells, what's the difference between starch and glycogen? Branching, degree of branching. Glycogen, very heavily branched. Starch, hardly branched at all. So starch usually is a mixture of amylose, which is not branched at all, and amylopectin, which has some branches. Now, when you make starch, do you have enzymes that add the branches? No. You make the linear oligosaccharide, and then there's what's called a branching enzyme that takes that and rearranges the bonds. You take a 1,4, you rearrange it to a 1,6. That's called a branching enzyme. Branching enzyme. So glycogen will use a UDP glucose synthetase. And bacteria, there are some that store glycogen. A few bacteria will use ADP glucose for glycogen synthesis. And then glycogen synthase will use UDP glucose as its substrate with the branching enzyme. Okay, so the concomitant activation of glycolysis and glycolysis would be very wasteful. That would constitute a futile cycle. So these two pathways show reciprocal regulation. Let's talk about that reciprocal regulation. First is the metabolite fructose 2,6-bisphosphate. 2,6-bisphosphate. Fructose 2,6-bisphosphate is a potent allosteric activator of PFK1. Potent allosteric activator of PFK1. But it's also a robust inhibitor of fructose 1,6-bisphosphatase. So this metabolite activates glycolysis, inhibits gluconeogenesis. So this is a pro-glycolysis metabolite. Pro-glycolysis metabolite. And this comes from your book. Notice that high levels of ATP actually, even though it's a substrate, they allosterically inhibit phosphofructokinase. So here is the activity of the enzyme with low ATP. Here's the activity of the enzyme with high ATP. I see a difference, do you? This activity is lower. It's lower. Does the activity go up as you increase the other substrate? Yes, but high activity allosterically inhibits the enzyme. So if the cell is producing more ATP than it can use, that slows down glycolysis. Now, if you were to take this reaction here and you were to add AMP or ADP, that would relieve the inhibition of ATP upon phosphofructokinase 1. So this event, this observation, can be reversed if you add AMP or ADP. That in the cell tells the cell you have sufficient quantities of uncharged adenosine phosphate molecules to be able to make more ATP. You need more ATP, so that will relieve that inhibition. Citrate. Now, citrate is made in the mitochondrion, but citrate is used to pass reducing elements, reducing equivalents to the cytosol during fatty acid synthesis. So if you have lots of citrate in the cytosol, that says that you're making fatty acids. You don't want to shunt your carbon then to another pathway under those conditions, correct? Well, if the cell makes more ATP than it needs, then you need to slow down glycolysis. Because what's glycolysis? It's an ATP making pathway. So if you have too much ATP, then you don't have enough AMP, ADP, you can't make more of it. So that essentially tells you things are going to grind to a halt. So before you get to that point, high ATP levels essentially slow down the enzyme. So you never really get to that point. So here is fructose 2,6-bisphosphate. Notice that ATP inhibits PFK1, AMP, ADP activated. Citrate inhibits it, and fructose 2,6-bisphosphate activated. These are the things that inhibit and activate phosphofructokinase 1.
Does it work all the time that what inhibits one direction, but the same one will?
Oh, not always, but many times, yes. So, yeah, you're being deep weeds. If you make mutations in PFK1 on the fruit fly, those animals die very early in embryonic development. In the mouse, the pups never form. They die as they get a maternal PFK1 message, and then they die in the second week, I think it is. Yeah, probably the same as humans. Yeah, there's a maternal contribution probably. In humans, that runs out very quickly. Humans are unusual in that their zygotic transcription starts very early, even earlier than the mouse. So the amount of maternal contribution is pretty minimal when it comes to how far it takes the embryo. So this actually very much gets to your question, because AMP, as you can see, where it activates phosphofructokinase 1, is an inhibitor of fructose 1,6-bisphosphatase. So in many cases, yes, and not in all cases. So it's not complete mirror image reciprocal, but it is in some. So ATP turns it off, ADP turns it on, AMP turns it on, citrate turns it off, and AMP does double duty. And if we're add to this fructose 2,6-bisphosphate, that would activate here and inhibit here. So that one would go straight across as well.
But it may not directly, for ADP, for example, it may not directly activate FBP, but the effect is kind of the same, right? If it activates the regular glycolysis cycle, then it's inhibiting the glycolysis cycle. So indirectly,
Probably because if the ATP charge is low, you're not going to be able to do gluconeogenesis. And since gluconeogenesis would be inhibited, then glycolysis kind of by default would be activated. So the answer to your question would probably be yes. Just because energetically gluconeogenesis is so expensive.
is there anything else that if it doesn't,
Yeah.
let's say it doesn't go through gluconeogenesis,
Yeah.
is there any other process, or are they?
Yeah, pentose phosphate pathway, which we'll get to another time. So, yeah.
Am I reading it correctly that the opposite, like after fructose 1,6-bisphosphate, is that what that means? I'm just confused about sugar.
No, this is the reaction here that's catalyzed by this enzyme.
Okay.
This is a reaction here that's catalyzed by this enzyme. These are the things that regulate these two reactions. This means it activates it. That means it inhibits it. That don't, yeah. So the presence, as you can see, of fructose 2,6-bisphosphate has pretty profound reciprocal consequences for fructose, phosphofructokinase 1 and fructose 1,6-bisphosphatase. So the presence of fructose 2,6-bisphosphatase is quite profoundly activating for PFK1. Without it, PFK1 is in pretty deep weeds. Without it, you need a lot of fructose 6 phosphate. This, as you can see, is going to decrease its requirement for this metabolite. However, with fructose 1,6 bisphosphatase, you can see the presence of this metabolite inhibits this enzyme. Without it, it's pretty active. With it, you need a lot of substrate before this thing even gets to be halfway active. Very, very potent inhibitor. Where here, it's a very potent activator. So fructose 2,6 bisphosphate is kind of the linchpin. That's whether or not you're going to do glycolysis or gluconeogenesis. And this essentially adds to this here. So fructose 2,6 bisphosphate activates glycolysis, inhibits gluconeogenesis, and it does it by either activating or inhibiting the central enzyme here of glycolysis, phosphofructokinase 1, and inhibiting fructose 1,6 bisphosphatase. So, from whence comes fructose 2,6 bisphosphate? It comes from phosphofructokinase 2. Phosphofructokinase 2, which takes fructose 6-phosphate, uses ATP, and makes fructose 2,6 bisphosphate. There's also a fructose 2,6 bisphosphatase, which is abbreviated FPPH2, which reconverts it to fructose 6-phosphate. So when this enzyme is active, you make fructose 2,6 bisphosphate, and glycolysis is encouraged, gluconeogenesis is discouraged. When you degrade fructose 2,6 bisphosphate, glycolysis is discouraged, and gluconeogenesis is encouraged. Now, yeah?
So fructose 2,6 bisphosphate has no other function besides encouraging.
It's right, it's a regulatory molecule.
Okay.
Now, here's the thing that's the brain twaddle. These two molecules are the same polypeptide. What determines whether it does one or the other is phosphorylation state. So when this protein is not phosphorylated, this is now PFK2. It will make fructose 2,6 bisphosphate. It will stimulate glycolysis and inhibit gluconeogenesis. When it's phosphorylated, now the fructose 2,6 bisphosphatase activity is active. The phosphofructokinase 2 activity is not active. You will now degrade fructose 2,6 bisphosphate. You will inhibit glycolysis, stimulate gluconeogenesis. What determines whether it's phosphorylated or not depends on hormones. So there's a phosphoprotein phosphatase. That phosphoprotein phosphatase takes off that phosphate. This protein is regulated by insulin. It's regulated by insulin. Insulin stimulates it. Whereas the kinase that phosphorylates it is the CAMP kinase, cyclic AMP-dependent protein kinase. This is activated by glucagon, because glucagon increases cyclic AMP levels inside the cell. That activates the cyclic AMP-dependent protein kinase, otherwise known as protein kinase A. That phosphorylates this enzyme, and now gluconeogenesis is encouraged, glycolysis is discouraged. And these two proteins have two separate domains. They're connected by a polypeptide link. And when you phosphorylate this protein, you phosphorylate it very close to the linkage. Essentially, that phosphate now has to find a little crevice. When it finds a crevice, the active site of the PFK1 now gets hidden. And the active site of the fructose 2,6-phosphatase is now wide open, ready to go. When you lop that off, now it changes conformation, and this one's active site gets hidden, and PFK2's active site is ready and raring to go. So it's the actual phosphorylation that determines which enzyme, which enzymatic activity is active in this polypeptide.
So it's two proteins, but one.
Right, 2 proteins on polypeptide.
How can that be two proteins, but one protein?
It's 2 activities, 2 protein activity, enzymatic activities in one polypeptide. So it's 2 protein enzymes, but they're the same polypeptide. But only one of those activities is active at a particular time.
So why do they not say it's inhibited or uninhibited by the phosphorylation? Sounds to me like, I mean, if it's the same polypeptide, it just depends on whether it's phosphorylated or not.
Right, then it has either it's phosphorylated as this activity.
Right,
If it's dephosphorylated as this activity.
right. But they've got 2 different meanings.
Yep.
It's the same polypeptide. It's 2 completely different.
But it's the same polypeptide.
Yeah. That's maybe more confusing.
Yep. If you clone this gene so that you only make this polypeptide or only make that one, they're active as monomers. They're active on their own. They're just not regulated.
So is that how insulin is that one way and how it decreases the glucose levels? Is it stimulating?
Yeah. Well, it's decreasing glucose levels actually has to do with an uptake protein called GLUT4. And the way that gets to the cell membrane is actually a very interesting story. I don't have time to go into it here, but that causes predominantly skeletal muscle fat and liver to take up glucose very, very quickly. That's what causes the blood glucose levels to come down rather precipitously.
That whatever side, the next one, right? The phosphate phosphatase leaves that phosphate group and it activates.
Right.
Yeah.
So now this particular activity is ascendant. This one is not. When it's dephosphorylated, this activity is ascendant. This one is not.
So it also wait, one more time. So PSK2 is when it is phosphorylated. It's not phosphorylated.
When it's not, when it's dephosphorylated. Dephosphorylated. All right. Have I blown your minds enough for one day? And it's not going to stop. So, hexokinase. Hexokinase. If glycolysis is not operating quickly enough, and you have a buildup of glucose 6-phosphate, hexokinase shows end product inhibition. So it's inhibited by glucose 6-phosphate. That's the case for hexokinase isozymes A, B, and C. Glucokinase is not. It is, however, inhibited by fructose 6-phosphate because that tells glucokinase that things are gummed up a little further down the pathway. And liver. It's hexokinase isozyme D. Glucokinase can be relieved of this inhibition by a protein in liver called the GRP, the glucokinase regulatory protein. That binds to glucokinase, relieves this inhibition. So if you have a lot of glucose coming into the liver, and liver needs to metabolize it quickly, and things aren't working quite fast enough, the glucokinase needs to keep phosphorylating that glucose, because if it doesn't, it'll seep out of the cell. This will bind to it. This is available when glucose levels are very high in the cell, and it'll say, okay, you keep phosphorylating away, and away it goes. Pyruvate kinase. This thing is really heavily regulated. Pyruvate kinase is inhibited allosterically by high levels of ATP. The ATP, if it gets too high, it decreases the affinity of the enzyme for this other substrate, phospholipyrate. Also, long-chain fatty acids will also inhibit it as well, because long-chain fatty acids tell you there is yet another carbon source that will actually give you more ATP per gram of carbon. That's beta oxidation, which occurs in the mitochondria. So if these build up, they're not being sent to the mitochondria fast enough, you shut down glycolysis. Pyruvate kinase comes in a couple of different isozymes. There's an L and an M version of pyruvate kinase. Just take a guess where the L version is expressed. Liver. Just take a guess where the M one is expressed. Muscle. Muscle. So the liver enzyme is subject to phosphorylation by protein kinase A. This again, protein kinase A is activated by cyclic AMP. Cyclic AMP is activated by G proteins. Glucagon is a G protein coupled receptor. So you get high cyclic AMP in the liver cell when glucagon binds. That will cause phosphorylation of the L-type enzyme that inactivates it. So when glucagon levels are high, pyruvate kinase is inactive. There's a protein phosphatase that will undo this. So as long as the glucagon is available, you'll keep inactivating it. As glucagon levels wane, now pyruvate kinase will start to be reactivated. Both liver and muscle pyruvate kinases are inhibited by ATP, activated by, activated by fructose 1,6-bisphosphate and inhibited by ATP, acetyl coenzyme A, long-chain fatty acids. ATP, acetyl coenzyme A, long-chain fatty acids. Okay, let's take a look at how hormones tweak the activity of some of these enzymes. So glycogen synthesis in muscle is a result of glycogen phosphorylase in muscle, but glycogen phosphorylase in muscle is slightly different than glycogen phosphorylase in liver. It's encoded by the same gene, but because of alternative splicing, you get a slightly different protein. So this slightly different protein at the amino terminus has a serine at position 14. That serine is a site for phosphorylation. This non-phosphorylated enzyme is not that active. It can work, but it's not all that active. So, if you activate phosphorylase kinase will phosphorylate that serine. And this phosphorylase is not quite active. I said this was for glycogen synthesis. I'm sorry. This is for glycogen degradation. Glycogen phosphorylates degrades glycogen. Yeah. Got that wrong. Yeah. Now, what stimulates glycogen breakdown? In liver, glucagon. In muscle, epinephrine. Both of these bind to G protein coupled receptors. Both of them increase cyclic AMP levels inside the cell. Cyclic AMP activates protein kinase A. Protein kinase A phosphorylates phosphorylase kinase. A phosphorylated phosphorylase kinase is now active. You have a phosphorylase kinase A and phosphorylase kinase B. The phosphorylase kinase B is inactive. If you phosphorylate it, it becomes the active form, phosphorylase kinase A, which is the active form. So this now becomes active. That targets then glycogen phosphorylase. This is now the active form of it. It degrades glycogen very quickly.
Would you mind if I say that one more a little slower?
This non-phosphorylated version of glycogen phosphorylase in liver, it's not active at all. In muscle, it's just a little bit active. Breakdown of glycogen gives you glucose for glycolysis, for fast energy. So if you want to degrade glucose, glycogen to make glucose for fast energy, epinephrine will help you do it in muscle. Epinephrine does not act in the liver. Glucagon does. Glucagon has virtually no activity in muscle. Glucagon will cause a breakdown of glycogen in liver, and it'll cause a release of glucose into the bloodstream to raise the blood glucose level. Glycogen that's broken down in the muscle stays in the muscle and is used by the muscle. So after this class where Emma's going to go to practice, he's going to do 5?
Today's an off week. I'm not actually running.
You're not actually running
I
today.
ran 3 yesterday.
You ran 3 yesterday, okay. After this class, he's going to dream about running, and if he was running, his muscles are going to need fast energy. His muscles have stored glycogen from lunch. As he gets going, that epinephrine that's going to be kicked in, that's an adaptation exercise, that's going to cause glycogen to be hydrolyzed in the muscle. That'll give his muscle energy because the epinephrine binds to the epinephrine receptor in the muscle, the beta 2 receptor. That increases cyclic AMP levels in the muscle. That activates protein kinase A, which phosphorylates phosphorylase kinase. And phosphorylase kinase will then phosphorylate as its target the glycogen phosphorylase. Now this becomes active. When it's active, it will degrade glycogen to glucose. The liver, however, is activated by glucagon. When do you make glucagon? In the post-absorptive state. So let's say you, who eats like a bird, you who has breakfast, and then if you get around to eating sometime later that day, maybe, just maybe, you'll have dinner. You didn't have breakfast either? Okay, point taken. You're in the post-absorptive state right now. Your liver is kicking out glucose to make sure your brain has enough glucose to keep you conscious. So that's degrading liver glycogen. And that's due to the fact that epinephrine doesn't affect liver, glucagon doesn't affect muscle. So these are two different tissues, but they have the same mechanism. That is, they activate phosphorylase kinase, and that degrades glycogen to glucose.
That's also kind of what you had.
Hang on.
So my brain is going to diabetes. I function. So when I exercise, my blood sugar will spike mid-exercise, but then it'll go back to normal. Is this why?
That depends on what have you eaten, how much insulin have you had, did you take insulin before you exercise, what was your sugar when you started? That depends on a lot of things. And every diabetic too is different.
Yeah, because I've always been told that when you exercise, if you spike, not to give
Yeah,
insulin.
yeah. Also, you're supposed to start a little high, and it'll go higher, but you shouldn't worry about it because it's going to gradually come down depending on how hard you're exercising.
I just always wondered what that's like.
Yeah. I had a student one year, they do light exercise, and they say, oh, I need to get to like 300 or 400 for this light exercise. I'm like, no. Who told you that? Whoever told you that's a dum-dum. Don't do that. And they called their doctor, and sure enough, I'm glad they didn't do that. But I had another one who was on the cross-country team. She'd started about 200. And that was perfect for her, because then, doing the P-loop a few times, it spiked probably 280, and then by the end of practice, she was probably at 120. So, which is a pretty big drop, but
Not for us.
no, it's not that bad. And plus 120 is a pretty good number for a diabetic. Then by then she'd eat dinner, and then she's where she needs to be, and then by the time she goes to bed, she'll give herself her nighttime insulin and then have a snack, and she'll be fine.
All of this
Yeah. Well, and this is why. This is why when you exercise, you'll spike.
and why would I get it done?
Yeah, precisely.
So then this is also kind of what you're talking about on Monday, how the liver is generous and the muscle is selfish. It
Yeah.
also keeps a lot of stuff
Yeah.
kind of present in the blood.
The liver's the Christian organ. Yeah. So the liver enzyme has a slight difference from the muscle enzyme, and that is it has an allosteric site for glucose. This serine also at the amino terminus has a tendency, when it's phosphorylated, to essentially tuck its amino terminus in like this, and these sites here, this particular molecule is active. When the glucose binds, now these sites essentially get exposed. So this is producing glucose as it degrades glycogen. But if that glucose starts to build up, that is, the liver cell can't transport it faster than it's making it, needs to slow down. So the glucose now binds, and you expose those serines. You expose those serines, and now they can be lopped off by the phosphorylase phosphatase. And that phosphorylase phosphatase will lop those off. This now is less active. So you're able to slow it down. This becomes a sensor, essentially, for glucose inside the cell.
That's an example of enzymatic inhibition.
It's not really inhibition. It's end product fine tuning, I suppose you could call it. All right, so during fasting, during the post-absorptive state, so let's say let's say you ate, let's say Divna back there ate breakfast at, I don't know, 7:30, and then say she skipped lunch. So she's in the post-absorptive state now. She's been making glycogen, a glucagon. And glucagon is made by the alpha cells of the pancreatic islets. Glucagon secretion is also downregulated by insulin. They tend to reciprocally regulate each other. That's why diabetics will have abnormally high levels of glucagon. And this binds to the G protein coupling receptor in the liver. It will then activate a trimeric G protein, on the inside of the cell. That then activates adenylate cyclase. That increases, now that it takes ATP, makes cyclic AMP. This will jack up cyclic AMP levels inside the cell. Cyclic AMP is a so-called second messenger. And then this second messenger is able to activate protein kinase A. Protein kinase A will then activate, will phosphorylate phosphorylase kinase, and it'll convert it to the active form. That will then activate glycogen phosphorylase. Then glycogen phosphorylase degrades glycogen to give you glucose. Why do you have these multi-step sort of activities? Well, with each step, you get amplification. So one occupied receptor might give you up to, say, 20 cyclic AMP molecules. Each of those cyclic AMP molecules will activate 10 protein kinase A's. That's a 200-fold amplification from the one hormone that bound here. Then activating phosphorylase kinase, one active protein kinase A can activate 100 phosphorylase kinase molecules. And then those can activate 1,000 glycogen phosphorylase molecules, which can then produce 10,000 glucose 1-phosphates. You can see here, you've got amplification all the way down. So what starts little then becomes big. That's the case. So remember, Jesus said, whoever is faithful in small things shall be also faithful in large things. A cell starts off with small things and becomes faithful in big things. There's a sermon illustration here. So cyclic AMP inside the cell is what's going to stimulate that glycogen breakdown. And that triggers protein kinase A, the CAMP kinase, you phosphorylate phosphorylase kinase, and then that's going to take the inactive form of glycogen phosphorylase and make it active. Then you break down glycogen to glucose 1-phosphate. But CAMP kinase also phosphorylates glycogen synthase. So this here, this is also going to phosphorylate glycogen synthase. Therefore, it shuts down the synthesis of glycogen at the same time. That makes sense. You don't want to be breaking it down and making it at the same time, do you? No. So you have essentially a process that prevents a futile cycle. Prevents a futile cycle. And then in muscle, epinephrine will also stimulate these events as well. And that's made by the adrenal medulla. Now, your cyclic AMP is going to activate CAMP kinase. CAMP kinase will phosphorylate the PFK2 FDPase2, the phosphofructokinase 2 fructose 2,6-bisphosphatase. So by phosphorylating that enzyme, you degrade fructose 2,6-bisphosphate. By degrading fructose 2,6-bisphosphate, you activate gluconeogenesis, and you inactivate glycolysis. This is how glucagon is going to activate gluconeogenesis and prevent glycolysis. And that's essentially how you regulate this thing called glycolysis. All right. Now, Friday, we get to start the Krebs cycle. Again, you must know every reaction, the structure of every intermediate, and every enzyme. And if we pay attention to the mechanism, you have to know the mechanism too. You also have to know how it's regulated. And that pathway is heavily regulated. Now, Krebs cycle also is a pathway that makes metabolites that are used for biosynthesis. So Krebs cycle intermediates get removed. So you have fill-in reactions called anaplerotic reactions that fill these in. There's a handful of those, you have to know those too. So Krebs cycle starts on Friday. Quiz, remember, is over chapter 14. And please watch a seminar. Yeah. And the gels are upstairs if you want to see them. They turned out, I thought, rather nicely. You did a good job. Go team. Next week, we will meet, not at 7:30, the morning group at least. Next week, we'll meet at 9. You'll take the ACS exam, and that's it. You don't need to clean up because Andrew's done all the cleanup. Everyone say thank you, Andrew. And then you just take the ACS exam, and then lab is done. Done. Internet assignment you do on your own. Yeah, do on your own. We're not meeting for that. It's due the week after the lab is due. Yeah. Okay. If you do better than you did last time, if your grade is on the knife edge, you get pushed over to the hallelujah side rather than the condemnation side. So it helps you, can only help you.
Okay.
Huh? 9 a.m., yeah. So get some sleep. Sorry? Just the morning section, afternoon section. We meet at the regular time. You take the ACS exam and then sayonara. If you want to, sure. Yeah. Sure. Yeah. If you want to come at 9 and you're in the afternoon section, please do. Yeah.